Good morning everyone, we are the Group 1 and in this video we would like to discuss and share our knowledge about DNA replications. What exactly is DNA replications? The purpose of DNA replication is to produce two identical copies of a DNA molecule. This is essential for cell division during growth or repair of damaged tissues. DNA replication ensures that each new cell receives its own copy of DNA. What problem can occur with DNA replication? DNA replication is a highly accurate process, but mistakes can occasionally occur, such as DNA polymerase inserting a wrong base. Uncorrected mistakes may sometimes lead to serious consequences such as cancer. Repair mechanisms correct the mistake.
DNA, like any other molecule, can undergo a variety of chemical reactions. Because DNA uniquely serves as a permanent copy of the cell genome, however, changes in its structure are of much greater consequence than are alterations in other cell components, such as RNAs or proteins. Mutations can result from the incorporation of incorrect bases during DNA replication. In addition, various chemical changes occur in DNA either spontaneously or as a result of exposure to chemicals or radiation. Such damage to DNA can block replication or transcription and can result in a high frequency of mutations, consequences that are unacceptable from the standpoint of cell reproduction. To maintain the integrity of the genomes, cells have therefore had to evolve mechanisms to repair damaged DNA. These mechanisms of DNA repair can be divided into two general classes. First, direct reversal of the chemical reaction responsible for DNA damage, and second, removal of the damaged bases followed by their replacement with newly synthesized DNA. Where DNA repair fails, additional mechanisms have evolved to enable cells to cope with the damage. Most damaged DNA is repaired by removal of the damaged bases, followed by the resynthesis of the excised region. Some lesions in DNA, however, can be repaired by direct reversal of the damage, which may be a more efficient way of dealing with specific types of DNA damage that occur frequently. Only a few types of DNA damage are repaired in this way, particularly pyrimidine dimers resulting from exposure to ultraviolet light and alkylated guanine residues that have been modified by the addition of methyl or ethyl groups at the O6 position of the purine ring. Excision repair. Although direct repair is an efficient way of dealing with particular types of DNA damage, excision repair is a more general means of repairing a wide variety of chemical alterations to DNA. Consequently, the various types of excision repair are the most important DNA repair mechanisms in both prokaryotic and eukaryotic cells. In excision repair, the damaged DNA is recognized and removed, either as free bases or as nucleotides. The resulting gap is then filled by synthesis of a new DNA strand, using the damaged complementary strand as a template. Three types of excision repair: base excision repair, nucleotide excision repair, and mismatch repair. Enables cells to cope with a variety of different kinds of DNA damage. Post-replication repair. The direct reversal and excision repair systems act to correct DNA damage before replication so that the replicative DNA synthesis can proceed using an undamaged DNA strand as a template. Should these systems fail, however, the cell has alternative mechanism for dealing with damaged DNA at the replication fork. Pyrimidine dimers and many other types of relations cannot be copied by the normal action of DNA polymerases, so replication is blocked at the sites of such damage. Downstream of the damaged site, however, replication can be initiated again by the synthesis of an Okazaki fragment and can proceed along the damaged template strand. The result is a daughter strand that has a gap opposite the site of damage to the parental strand. One of the types of mechanisms may be used to repair such gaps in newly synthesized DNA: recombinational repair or error-prone repair.